Gas vesicle nanoparticles, or GVMPs, are a type of prokaryotic organelle, meaning they are an organized structure found in some bacterial and archaeal cells. GVMPs are unique because they are enclosed in a membrane made entirely out of proteins. There are over a dozen proteins involved in GVMP synthesis, but the main structural proteins are GVPA, which forms the body of the organelle, and GVPC, which attaches to the outside of the organelle along GVPA's spiral ribbon. In addition to being a unique cellular structure, GVMPs can be engineered to serve a variety of potential medical applications using fusion proteins. Fusion proteins are created when you take one segment of DNA that codes for one type of protein and another segment of DNA that contains the instructions for another protein, and you sever them in specific places and splice them together to create one recombinant DNA molecule. This molecule can be used to print out fusion proteins, and if you do it right, these fusion proteins will have properties from both of the original proteins. You can do this with the GVPC in GVMPs to create GVPC fusion proteins, which attach to GVPA's spiral ribbon and allow GVMPs to display a range of different proteins for different functions. For instance, you can fuse GVPC with viral antigens to use in vaccines. These antigens are the parts of viruses that allow our antibodies to recognize and attack them. Attaching the antigens to GVMPs stabilizes them and allows vaccines to become more effective, while the non-toxic nature of GVMPs decreases vaccines' reactogenicity, which is the normal adverse reactions to vaccination. You can also attach GVPC to something called immunoglobulin-binding streptococcal protein G, which is the protein that attaches almost all antibodies to their cells. This results in GVMPs that can display antibodies, allowing them to be used in treating diseases such as autoimmune disorders or cancer. One of the reasons that GVMPs are so well suited for these purposes is that they are incredibly stable, allowing them to extend these products' shelf life for months and be stored without the need for an expensive and potentially unreliable cold chain. Unfortunately, GVMPs are so stable that scientists haven't been able to fully take them apart, so no medications using them can make it to clinical trials until we know exactly what's inside them. That being said, GVMPs have been shown to be non-toxic to animals, and they have almost unlimited potential for biomedical applications.